Hi, hello, good morning everyone. I'm Dr. Harika Kishore. On behalf of Vista's Learning, I'm here as your biology educator. V-Learning, it's an online education platform which works with an ideology of providing quality education for all. Visit our website www.v-learning.in where you're going to get an access for all the live classes, recorded sessions, study materials, concept videos. Moreover, you're going to get this lecture notes in the form of PDFs. And also make sure to get your acquaintance with all the channels available and subscribe to the channel Vista's Learning Class 11 and 12. If you find this session useful and interesting, click on the like and share this session with your friends. And also click on the bell icon so that you are going to get all the upcoming video notifications. Saying that, let's begin our today's session. We are in the series of the session where we are discussing about the chapter Molecular Biology of Inheritance that comes to the unit Genetics and Evolution, right? Now, so in the previous session, we have discussed about the salient features of a double helical structure of DNA that was proposed by the Watson and Crick. After understanding the structure, characteristic features of the double helical structure of the DNA, let's move on to the next topic, and that is the search for genetic material. Now, by now we came to understand that whatever the characteristics or the traits that are coming from one generation to the another generation that are carried by the genetic material. Now, today's session, we are going to understand the biochemical nature of that genetic material which carries the information to make a whole organism, right? Now, so here, even though the discovery of nuclein, any substances, whether it is protein, nucleic acids like DNA, RNA, which is present inside the nucleus, we call it as a nuclein, and that was discovered by Mescher. And the proposition for principles of inheritance which were given by the Mendelian laws of inheritance were almost discovered at the same time. But that the DNA acts as a genetic material took long time to be discovered and proven. By now, we know that DNA is going to be act as a genetic material. But then it was not known, it was not clear that the DNA is the biochemical nature of this genetic material. So here, that is by 1926, the quest, search to determine the mechanism for genetic inheritance. Mechanism for the genetic inheritance had reached molecular level. Molecular level, we can say that are very minor details. Cellular level, inside the cell, we have molecules. And the study reached to the molecular level by 1926. Now, previous discoveries like so many scientists, biologists and geneticists have done their research, and list goes like Gregor John Mendel, Walter Sutton, Boveri Sutton, famous, known for their discovery, and even Thomas Hunt Morgan. These are all the scientists who have worked about the importance of the genetic material, and these genetic material may be present inside the nucleus at the chromosomal level. So here, and numerous other scientists had narrowed down. However, our main motto is to understand the genetic material and its biochemical nature, where it is present, whether it is present inside the cell or it is present in the nucleus. In the nucleus, we have DNA, RNA and protein, in which the molecule is present that is going to act as a genetic material. So now, has narrowed down the search to the chromosome. This search has reached that the genetic material is present in the cell. Now it has been narrowed down such that the genetic material is present inside the nucleus of the cell. That means it is narrowed down at the chromosome which is present inside the nucleus, that is located in the nucleus of the most cells, right? But the question here, what molecule was actually acting as a genetic material? That was not answered yet. Whether the protein is acting as a genetic material, whether the DNA, whether that is RNA. We don't know. The genetic material may be present inside the nucleus of the cell, and all the experiments which were conducted by this set of scientists have narrowed down to the chromosome. But inside the nucleus also, we do have the presence of the protein, DNA and RNA. Out of these molecules, which is the biochemical nature of the genetic material. Now, so here, in 1928, Frederick Griffith done a series of the experiments. In a series of experiments, what he did by using a bacterium, and that bacterium is Streptococcus pneumoniae. It's a bacteria which is responsible to cause pneumonia. Now, witnessed a miracle and miraculous transformation is happening in the bacterium, and that bacterium is Streptococcus pneumoniae. So here, during the course of its experiment, he have done a series of experiments in which a living organism, that is bacterium, nothing but Streptococcus pneumoniae, that is bacterium, has changed its physical form. Now, so here, when Streptococcus pneumoniae, pneumococcus bacteria are grown in a culture, we have taken a petri dishes which is having the medium, nutrient medium, and we started growing this Streptococcus pneumoniae bacteria. Now, we find that some of the bacteriums are smooth and some other are rough. Some of the bacterium looks very smooth and shiny because it is having a polysaccharide coat around it, and because of that it looks very smooth. So we call this strain as S strain, S for smooth. And other bacterium are rough in nature, they are not smooth because they don't have that mucus polysaccharide coat around them. Therefore, this colony is full of rough bacterium, rough strain. That is what we represent that strain as R strain. So this is the S strain and that is R strain. So here, due to the presence of this polysaccharide coat, mucus polysaccharide coat, it is stable against the immunity of an host organism. And whereas these strains do not have the polysaccharide coat, because of that reason they are avirulent. So S forms are smooth, shiny, they contain polysaccharide coat and they are virulent form. Virulent means they are capable of causing pneumonia disease. Coming to the R strain, they are rough, they don't have polysaccharide coat, and therefore they can't cause the infection and they can't cause pneumonia in the host organisms. So here, some produces smooth shiny colonies, what we call it as a S strain, while other produces rough colony we represent as a R strain. Now, taking these two strains, Griffith conducted a series of experiments by using laboratory animal, that is mice. So here, this is because the S strain is smooth, because as we said that it contains polysaccharide coat, because the bacteria has a mucus polysaccharide coat, while the R strain do not have that polysaccharide coat. That is why it looks rough. That is what we given as a rough strain or R strain. Now, when mice is infected with a S strain, as we already said, it has a polysaccharide coat and it is stable towards the immunity of the mice. Therefore, it is virulent. See here, mice infected with S strain, as the S strain is virulent, mice is infected those which are infected with the S strain die from pneumonia infection. But the mice infected with the R strain do not develop any pneumonia and they are live. So let's first understand about series of the experiments which were performed by the Griffiths. So in the first experiment, he have taken S strain and he injected this S strain into mice. As this is virulent form, it causes pneumonia which leads to death of the mice, that is, mice die. Now, second, he have selected R strain. Now, the same R strains were used to infect mice, as the R strains do not contain polysaccharide coat. They are not stable towards the immune system of the mice. Therefore, they can't produce the disease, infection, pneumonia. Therefore, the mice is live. Now, in the next series of experiment, he have selected S strain, but this S strain were heat killed. He subjected the S strains to the heat treatment. Therefore, the heat killed S strains were produced. Now, the same thing, he injected heat killed S strain. Now, pneumonia is not formed, therefore the mice is live. Whereas its live form, that means those strains which are not given heat treatment, they can cause pneumonia and which leads to the death of the mice. But here, as the heat treatment is given to the S strain, now these S strains are not capable of producing the pneumonia, therefore the mice is live. Now, in the fourth series of experiments, where he have taken heat killed S strain plus live R strain. In both cases individually, where heat killed S strain is not capable of producing pneumonia, whereas the R strain are also not capable of producing pneumonia. In both the cases, the mice are live. But here the combination, when he used to infect and inject into this mice, to his surprise, the death of the mice took place. The reason here, so here the simple thing, 4 series of experiments, where in the first series he have taken S strain, injected into mice, pneumonia produced, mice die. Now, R strain, injected into mice, pneumonia is not produced, mice is live. Next, he have taken the S strain but given the heat treatment. So, heat killed S strain injected into mice is not able to produce pneumonia, mice is live. Now, he combined what? He combined this with R strain. See, heat killed S strain plus live R strain. Individually, both will not cause pneumonia and the mice is live. But in combination, it is able to cause pneumonia and causes the death of the mice. Here, coming to the conclusions of this experiment, this is simple, where Griffith was able to kill bacterium by heating them. That is what we call it as a S strain heat killed. Right? So, he observed that the heat killed S strain bacteria injected into mice, it cannot produce a pneumonia, therefore the mice didn't kill. Now, when he injected a mixture of heat-killed S strain as well as live R bacterium, it is able to cause pneumonia and leads to the death of the mice. So here, moreover, he recovered from this dead mice. Griffith is able to recover the living S bacterium. The question is, to this mice, he have injected heat-killed S type and live R type. But he could able to recover live S type, where it is coming from. For this, he stated that transformation happened. Transformation happened from the heat-killed S strain to the R strain, making the R strain into virulent form. And he named that as a transforming principle. So he concluded that R strain bacterium had somehow been transformed, that is what transforming principle, transformed by the heat-killed S strain bacterium. When he combined the S strain heat-killed S strain bacterium with live R strain, some transformation happens from heat-killed S strain to live R strain, which makes the virulent, non-virulent R strain into virulent form. Some transforming principle transferred from the heat-killed S strain has enabled the live R strain to synthesize a smooth polysaccharide coat, which is not usually present in the R strains. That is what we have given a rough strain, R strain name, right? And become virulent. So that is how, after some transformation has been happened from S strain heat kill to the live R strains, now the live R strain become virulent, which is able to cause the disease. For this, he named the transforming principle. Now, this must be due to the transfer of genetic material. The R strain got the capacity to produce polysaccharide code. It can now produce polysaccharide code. That is how it become virulent. And this is all happened because of the transfer of genetic material to R strain. And that genetic material will give the information which is needed for the synthesis of the polysaccharide coat around it and making this R strain virulent form. Now, however, the biochemical nature of this genetic material, yes, correct. Some transfer of the genetic material takes place. Because of that, the heat-killed S type will able to transfer its information to the R strain, making the R strain convert into virulent form. But whatever the transfer genetic material, its biochemical nature, whether it is a protein, whether it is a RNA, whether it is a DNA, was not defined from his experiment. He defined that some transforming principle is happening in such a way that it got the new information to synthesize the polysaccharide coat around it and making the R strain into virulent form. Now, prior to the work of Oswald Avery, Colin MacLeod and Macklin McCarthy, very popular, called as Avery-MacLeod-McCarthy experiment. Avery-MacLeod-McCarthy experiment, who did their experiment during 1933 to 44. The genetic material was thought to be at that time out of protein, DNA and RNA. The genetic material at that time, it was thought protein is going to act as genetic material. So they worked to determine the biochemical nature of the transforming principle. We know that this transforming principle, it transforms the genetic material. And that genetic material was thought to be protein. So, now, how they are going to prove that that is a protein? But the purified biochemicals like protein, DNA, RNA from the heat-killed S cells to see which one could transform the live live R cells into S cells. That means R strain to S strain. So here, they discovered the DNA alone from S bacterium causes the R bacterium to become transformed. So, towards the starting of the experiment, before starting the experiment, prior to the experiment, they thought like every McLeod, they thought like the genetic material, maybe it is protein. And they started working on it and discovered that the protein is not the genetic material, that is the biochemical nature of the genetic material that is making this R strain into S strain, but that is DNA, because DNA can alone is fine to be transformed. Now, so here for that to confirm what they did, how they confirm that protein is not the genetic material and the DNA is the genetic material. For that, what they have done, they did their experiment by using enzymes. They also discovered the protein digesting enzymes, proteases, and RNA digesting enzyme, RNases, did not affect the transformation. See, first they have taken proteases, RNases, and DNase. We know that these are the three enzymes. Proteases can break down the proteins. DNases, RNases can break down the RNA. DNase can break down the DNA, right? So, in a mixture, they have taken protein, DNA, RNA. Then they added proteases, right? If the proteases was added to this mixture, proteins are going to break down, right? It is going to break down. What are the molecules left out? DNA and RNA. When the protein, DNA, RNA mixture, which is subjected to the proteases and proceeds to the transformation experiment was successful. Here, transformation happened. That means in the absence of protein also transformation is happening, which means protein is not responsible for the transformation to be happened. Now, after that, they have provided RNases enzyme and proceeds to the transformation experiment. If the RNase enzyme is present, it is going to break RNA. Now, RNA is not present, but still transformation done, took place. That means transformation happens successfully. In the absence of protein and in the absence of the RNA, even the transformation is happening, means for the transformation to happen, RNA as well as protein are not responsible. Which is remaining? DNA. Now the DNA, if it is DNases, if it is providing, which breaks the DNA, then the transformation is not happened. That means the transformation to happen, the presence of the DNA is important. Therefore, they come to the conclusion that genetic material is DNA. So here, digestion with DNases, finally they have added DNase enzyme, which can break the DNA. Digestion with DNases did inhibit transformation. After addition of the DNases enzyme, the transformation doesn't happen because it is inhibited. suggesting that DNA caused the transformation. That means transformation to occur, DNA is responsible, and therefore this DNA is acting as a genetic material. Right? They concluded that DNA is the hereditary material, genetic material, but not all the biologists were convinced by this experiments. There's still more experiments to convince that the DNA is acting as a genetic material, but not the RNA or protein. Then another experiments, very undoubtable proof, unequivocal. The unequivocal proof that DNA is genetic material came from the experiments of Hashi and Chase, which was popularly called as Hashi and Chase experiment. Alfred Hashi and Martha Chase. They did their experiment in the year 1952. Say before that, they were experiments which were done by the Avery McLeod experiment by taking the enzymes, proteases, DNases, RNases. They concluded that DNA is a genetic material, but it was further confirmed by Hashi and Chase experiments. What is that tremendous experiment that proved that DNA is a genetic material? Let's see. They worked with the viruses, and that virus is bacteriophage. Bacteriophage. It is a virus which can infect bacteria. Because of that, the name bacteriophage, which literally means bacteria eating. This virus can eat the bacteria. Therefore, it got its name bacteriophage. So, they have taken this bacteriophage, which is able to infect bacteria, and that is what we call it as a bacteriophage, right? If you remember the structure of the bacteriophage, so it will have head part, it will have tail part, and this is head. This is collar domain, and this is sheath. This is base plate, and these are tail fibers, right? So this is the typical structure of the bacteriophage, which can infect the bacterium. So the bacteriophage first attaches to the bacterium, and then it will inject its genetic material inside the bacterium, and its genetic material then enters the bacterial cell. The bacterial cell treats that genetic material. See, for example, this is the bacterium which has been infected with your bacteriophage. It injected its genetic material. That means the virus genetic material is now inside the bacterium. This bacterium will treat this genetic material of the virus as its own. The genetic material as if it was its own. It means here bacteria. Bacteria will treat that genetic material as its own and start subsequently manufactures more viral particles. So here what Hashi and Chase worked to discover whether it was protein or DNA. They have a doubt between whether the genetic material is protein or the genetic material is DNA. For that, they have selected the bacteriophages, which is able to infect the bacterium. By doing the experiment, they came to the conclusion that DNA is going to act as a genetic material from the virus that enters the bacterium. See here, just now we said that the virus right? This is the virus. After attaching to the bacterial surface, bacterial cell wall, it is going to inject its genetic material. Now the question is whether this genetic material is DNA or protein, because the virus is able to infect this bacteria by pumping definitely its genetic material. Now we are interested to know the biochemical nature of that genetic material, whether it is protein or whether it is DNA. So here they grow this viruses, that is bacteriophages, on a medium that contains radioactive phosphorus as well as radioactive sulfur. They have taken 2 petri dishes, and in these 2 petri dishes, it have nutrient medium which supports the growth of the bacteriophages. But here, one medium is provided with the radioactive phosphorus and the other medium is provided with the radioactive sulfur. So, whatever the bacteriophages that are growing in this plate will be radioactive phosphorus labeled bacteriophages, whereas here it will get the radioactive sulfur. Now, we already know DNA in its structure, double helical structure, right? The backbone is made up of phosphate, sugar, phosphate, sugar, phosphate backbone. That means DNA have phosphorus. Just now we said that one plate is labeled radioactive phosphorus. So this radioactive phosphorus will enter into DNA of the viruses. Whereas coming to the sulfur, if you take the structure of the protein is made up of amino acids. And the amino acids in its side chain will have sulfur. Like methionine, cysteine, these are all going to have the sulfur in it. That means the sulfur is present in protein. Therefore, whatever the bacteriophages that are growing in the medium that is containing radioactive sulfur will go and sit to the protein part of the bacteriophage. Here, the radioactive phosphorus enters into DNA and the radioactive sulfur will enter into protein. Here, the radioactive phosphorus cannot radiolabel protein content because in the protein we don't find phosphorus. In the same way, the radioactive sulfur will not attach to the DNA because throughout the structure of the DNA we don't find the sulfur to be present, right? So here, virus grown in the presence of radioactive phosphorus contain radioactive DNA. So here we got radioactive DNA because phosphorus will be present in the DNA, and here we'll get radioactive protein because sulfur will not be present in the DNA, but not radioactive protein. Here we'll get only radioactive DNA. Because DNA contains phosphorus, but protein do not. What protein have? They have sulfur. Similarly, virus growing on radioactive sulfur contained radioactive protein, but not radioactive DNA because DNA does not contain sulfur. So we have taken two petri dishes. One is provided with a radioactive phosphorus, which forms radioactive DNA, and the other petri plate have radioactive sulfur, which forms radioactive protein. That's it. Now, coming to here. So this is bacteriophase, right? We have taken two petri dishes where we'll get here protein is labeled. So that is what? Radioactive protein. Protein, that is sulfur. Sulfur 35. Here we got radioactive DNA, that is phosphorus 32. We have two types of bacteriophages now. In the first set, the protein part of the bacteriophage is radioactive, whereas in the second set, we have the DNA part of the bacteriophage is radio labeled. That is what? Radioactive DNA, radioactive protein. Just now we have seen the structure. Inside the head, it has genetic material. Inside the head, it has genetic material. Now, this genetic material is DNA and this is protein. Now, the question is whether which is entering into bacterial cell, whether that is protein or DNA. Now, here in this, the outer protein part is radio label. It is represented by the red color. And here the DNA is radio label, which is represented with the red color. Now both the bacteriophages are allowed to infect the bacterium. Now we know that the life cycle of the bacteriophages, where after infecting the bacterium, it go and first attaches to the cell surface of the bacterium. That stage is called as absorption. Once the absorption was successful, then it starts penetrating. Penetrating what? It injects its genetic material to enter into bacteria. Here, see, after absorption, penetration starts, where the virus is injecting its genetic material. The same thing happens here. Now, once the genetic material, just like a syringe, once the liquid is passed into the bacterium, that means the genetic material is injected by the viruses, the outside protein coat, we call it as this is protein coat. Protein coat. Now this protein coat will be present outside the bacterial cell itself. See, here also the protein coat is remaining outside the bacterial cell, which enters the bacterial cell. The genetic material of the virus is injected into the bacterial cell. Now after this protein coats gets detached from the bacterial cell. At this stage, what we will do here is the infection is happening, where the virus starts infecting the bacteria by injecting its genetic material into the bacterium. Now once that stage is finished, now we'll blend this one such that we'll get the protein coat as well as the bacterial cell. After subjecting to the centrifugation, centrifugation is a separation method where it will be separated into pellet and supernatant. Pellet will be deposited, sedimented, and the supernatant will come to the top layer. So here, see, we have two things in the centrifugation mixture. We have protein coat of the viruses and we have the bacterial cell. Now, the genetic material is inside the bacteria, but we are interested to know whether that is protein or DNA. To identify that, we are given two radioactive substances. The protein is labeled with the sulfur 35 and DNA is labeled with the phosphorus 32. Now here, once the centrifugation is happened, we can see no radioactive sulfur 35 detected in the cell, because here the protein is labeled and the protein is detached from the bacterial cell. Inside the bacterial cell, we don't have sulfur 35, S35, and the radioactive sulfur 35 is present in the supernatant, because it is not in the inside the cell. The bacterial cells goes to the pellet and the supernatant is filled with the protein coat. Coming to the same centrifugation that is happened in this case, where we find radio label phosphorus 32 that to be present inside the bacterial cells, detected in the cells, and the supernatant will be protein coat. Protein coat is labeled with the no labeling, therefore no radioactivity is detected in the supernatant, because here the protein coat is not labeled, that means radio labeled. So by this experiments, Hashi and Chase concluded that the genetic material is DNA but not protein. So now, radioactive phases, we have two. One is radioactive protein and the one is radioactive DNA. We're allowed to attach to the Escherichia coli, there is a bacterium. Then as the infection proceeds, the viral coat, that is a protein coat, were removed from the bacterium by agitating them in a blender and now subjected to centrifugation. The viral particles were separated from the bacterium by spinning them in a centrifuge. That is what centrifugation happens. So inside the bacterial cell, what is present? That is genetic material. That is important for us. Bacteria which was infected with the virus that had radioactive DNA. Radioactive DNA were radioactive, indicating that the DNA was the material, that is a genetic material which passes from the virus to the bacterium, which passes inside the bacterium. Genetic material is DNA but not protein. This is the conclusion of this experiments. Bacteria that were infected with the viruses that had radioactive protein were not radio labeled because it is present in the supernatant. This indicates that protein did not enter the bacterium from the viruses. That means the protein is present outside itself. It is not entering into the bacterium. That means it is not going to act as a genetic material. DNA is therefore the genetic material that is passed from virus to bacteria. So this is what we explained. So here both the radioactive protein and the radioactive DNA labeled bacteriophages were allowed to infect the bacterium, and once the infection is done, it is blended, it is centrifuged and looked for the DNA active, that means radioactive substances which is present inside the bacterium. If you look inside the bacterium, it is not radio labeled. If you look inside the bacterium, it is radio labeled, and that radio labeled substance is phosphorus 32. Phosphorus 32 will get radio label DNA only, but not protein, right? Yeah, with the series of the experiments, it was concluded that the genetic material is DNA, but not RNA or protein. That is it for the today's session. We meet in the next session. Until then, keep learning with We Learning. Thank you.